Good evening, everyone. In today's session, we will discuss about chapter 19, that is excretory products and its elimination from class 11th biology of NCERT. So first of all, why these excretory products are present in our body? These products are either the byproducts of metabolic processes or they are excreted when the ingestion is in higher amount. But mostly they are the byproducts of the metabolic activities. To maintain the homeostasis of the body, these products are released out
अभी
of
भी
the
कुछ
body.
बैकग्राउंड में घरघराहट आ रहा, क्या वायर है क्या आपका कान में लगा हुआ है कुछ?
हाँ शायद सर वायर है इसलिए.
उसको थोड़ा सा उसको कर लो, स्टैटिक कर लो.
ओके सर,
में
हो
आ रहा
गया.
है ना, उसको कर दो. हाँ,
Okay. So these products are either the byproducts of the metabolic waste processes, or they can be excreted because there is accumulation of uh, these byproducts. Okay, so the animal accumulates ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, chloride, phosphate, and sulfate uh, in their body, and that's why they has to be removed out of the body. There are three common nitrogen wastes, that are ammonia, urea, and uric acid. The process of release of ammonia is called as ammonotelism. When urea is released, the process is known as ureotelism, and when uric acid is released, the process is called as uricotelism. And out of these three nitrogenous wastes, the ammonia is most toxic form of the excreted product, and whereas urea and uric acid are the less toxic products. Now ammonia are released in ammonium ions. They require a large amount of water for their elimination because it is very toxic. That's why it requires a large amount of water for its excretion, because this toxic compound cannot be present in the concentrated form inside the body of the organisms, as it is also readily soluble in water, so it is very easy to dissolve it in the water. The organisms that release ammonia are called as ammonotelic animals, and these animals are bonifacious, aquatic amphibians, and aquatic insects. So these animals do not have any requirement to store water or to conserve water, because they have plenty of water around them, so they can easily remove large amount of water from their body. And sometimes the ammonia is diffused from the body surfaces, or they can be released through the gill surfaces in fishes. So here there is a significant role of the kidneys, but the organism, like the terrestrial organisms, they adapt for the conservation of water. So the ammonia is converted into the less toxic product like urea and uric acid for the conservation of water. Now urea. The animals release urea as the excretory product are called as ureotelic animals, and these animals are mammals, many terrestrial amphibians, and marine fishes. They require the water conservation for their body. That's why in liver the ammonia is converted into urea by the urea cycle, and then it is transported to the kidney via the blood, and then from the kidney it is filtered out from the body. The some amount of urea may be retained in the kidney matrix, and here it is very important that the urea should be retained in the kidney to maintain the osmolarity in the kidney. The next is the uric acid. So the animal release uric acid are known as uricotelic animals. These are reptiles, birds, land snails, and insects. So these animals cannot afford to release a very large amount of water, so very small amount of water is released. That's why the uric acid is released in the form of pellet or paste, and it is also least toxic. But the conversion of ammonia into the uric acid require a very large amount of energy, but for the conservation of water is very necessary here, so ammonia is uh, converted into the uric acid. Now we will discuss the excretory structures which are important for the release of these excretory products. So in invertebrates, the excretory structures that are very very simple and in tubular form, but in the vertebrates the excretory structures are very complex and like the kidneys. The primary function of these excretory structures is to maintain the ionic as well as the fluid volume regulation uh, for. Okay, so the first is protonephridia or flame cells. So here is the example of the flame cell. So a bundle of flame cells are called as protonephridia, and it is a single flame cell. So this it is found in the platyhelminthes, flatworms, example planaria, rotifort, some annelids, and the cephalochordate like amphioxus. These flame cells have a cluster of cilia. Okay, so it the cilia moves the excretory product inside the cell. The cell have a nucleus. Okay, it moves in the cell because the it has a blind ended tubules. So the excretory products are first uh, first excreted inside the cell, and with the help of the tubes they are removed out of the body. The next is the nephridia. Nephridia are found in the earthworm and other annelids. These have the same mechanism like the protonephridia, but in the flame cell, as first the excretory products are released inside the cell, in case of nephridia, the excretory products are directly move outside the body. Okay, so uh, there is no accumulation of the excretory product inside the body. The next is the Malpighian tubules. These are found in most of the insects and like in cockroaches. So in this diagram, we can see this is the alimentary canal. The alimentary canal is present in the hemocoel cavity of the cockroach, and this cavity have a fluid that is called as hemolymph. Okay, so in this alimentary canal, this is the midgut, and this is the hindgut. So between the midgut and hindgut, Malpighian tubules are present. These Malpighian tubules are made up of single layer of cells. The This single layer of cells absorb what solutes, water, and waste from the hemolymph. Okay, and then it secrete all of it into the colon. Okay, so the important constituents like the water and the solutes, they are again absorbed from the colon, but the waste product is released out of the body in form of thick paste or powder. The next is antennal glands. So in the organisms like crustaceans, like prawns, these antennal glands are present. These antennal glands are present in each side of the head region. So this head region here and here antennal glands are present, and like the kidney, the blood is filtered in the end sac, and then with the help of this tube the excretory product is moved to the bladder and then released out of the body. Now we will discuss the human excretory system. So the human excretory system contain a pair of kidney, a pair of kidney, a pair of ureter, then the urinary bladder, and urethra. So here, the right kidney is little bit in the little bit downward from the left kidney, because above the left kidney liver is present. Okay, so that's why it did not get enough space to uh, uh, to present at the same level of the left kidney. So the first is kidney. So kidney is a retroperitoneal organ. That means it is posterior to the peritoneum. Peritoneum is the layer which surround the abdominal cavity. So the kidney is present posterior to this peritoneum. That's why it is called as retroperitoneal organ. And this is the anterior view of the kidney, that how it is looks like. Now kidney is a reddish brown and bean shaped structure which is situated in the last thoracic and third lumbar vertebra, that is close to the dorsal inner wall of the abdominal cavity. Each kidney of the adult measures length is 10 to 12 centimeter, width is 5 to 7 centimeter, thickness is 2 to 3 centimeter, and each kidney weigh approximately 120 to 170 gram. Now the structure of the kidney. The whole kidney is divided into the two sections. The one is the outer section is called as the cortex, and the inner section is divided in the form of the medullary pyramids. Okay, and the whole kidney has a covered by a tough layer that is called as renal capsule that protect the kidney from any type of injury. Between these medullary pyramids, the cortex is extended and form columns. These columns are called as renal column or column of Bertini. Then the medullary pyramids are extended to the calyx, form the calyx. The number of calyx are then joined to form the renal pelvis, and then it is connected to the ureter. Okay, so here is a concave uh, structure uh, that gives the entry to the linear artery, renal vein, and nerves to that enters inside the kidney. The next is the functions of the kidney. So apart from the excretion of wastes, there are number of functions of the kidney. Okay, so first is the excretion of the wastes. As we all know, it helps in the excretion of urine. The next is the regulation of the blood ionic composition. So by the blood, it absorb and secrete a number of ions, and hence it regulates the ionic composition of the blood. The next is the regulation of blood pH. So the blood pH is maintained with the help of the bicarbonate buffer system, through which the homeostasis of the whole body is maintained. The next is the regulation of the blood volume. Okay, so when there is a drop in the blood volume, so it is required to conserve water. So for conservation of water, the kidney will produce a concentrated urine, and hence it uh, conserve water. And if there is increase in the blood volume, there is elimination of water, and hence the kidney will form dilute urine. Okay, so it has its own way to conserve and eliminate water for the regulation of blood volume. Next is the regulation of the blood pressure. Okay, so the blood pressure is very important in the formation of urine, and it is done with the help of the secretion of the enzyme like renin. Renin play a very important role in the maintenance of the blood pressure in the kidney by uh, by activating number of hormones, and hence it also regulate the filtration of blood. Next is the maintenance of blood osmolarity. So the osmolarity is basically directly proportional to the concentration of the solute present. If the solute concentration is more, the osmolarity will also be more. Okay, so if there is loss of solute, then there should also be loss of water to maintain the osmolarity, and it is done by the kidney. The next is the production of hormones. There are two types of hormones that are also not important for the excretion purpose, but they are produced by the kidney. One is the calcitrol, which is the active form of the vitamin D that helps in the regulation of calcium homeostasis, and the other hormone, which is known as erythropoietin, that stimulate the production of the red blood cell. That means it stimulate the production of the RBCs, regulation of the blood glucose level. So, there it is very important to regulate the blood glucose level. And when there is decrease in the blood glucose level, there is a process that is known as gluconeogenesis, which is primarily occur in the liver, but kidney or some amount of gluconeogenesis also occur in the kidney. In this process, the glucose is formed from the non-carbohydrate source. Okay, so that's how the low level of glucose in the blood can be increased with the help of the gluconeogenesis. The next is the kidney contain a number of each kidney have 1 million complex tubular structure that is known as nephrons, and hence they are known as the functional unit because they play a very important role in the formation of the urine. Okay, so what is the path of the urine drainage? Okay, so this is the nephron. Okay, so as we see in this picture, so and this is the collecting duct. So there are number of nephrons present here, and all the nephrons are connected to the collecting duct, and then the is drained into the collecting duct. The collecting duct then drain urine in the minor calyx. These are the okay. So these are the minor calyx. The then they and the urine is entered into the major calyx. Then all the major calyx drain it into the renal pelvis, and finally the renal pelvis is transport the urine to the ureter, and then it is stored in the urinary bladder. So there are two major part of the nephron. The one is the glomerulus and the next is the renal tubule. So first we will discuss about the glomerulus. So what is the glomerulus? Glomerulus is basically a ball-like structure which has a number of capillaries. Okay, so it has an afferent arteriole and efferent arteriole. Afferent arteriole means the blood supply uh, take bloods towards the glomerulus, and when the blood is taken away from the glomerulus, it is called as efferent arteriole. The glomerulus is surrounded by a capsule-like structure which is called as Bowman's capsule, and collectively the glomerulus and Bowman's capsule are called as Malpighian body or renal corpuscles. The Bowman's capsule then further extended to the proximal convoluted tubule, which we will discuss later. Okay, so this is the other parts of the nephrons. So as we discuss about the glomerulus and Bowman's capsule, now the Bowman's capsule is further extended to form the proximal convoluted tubule. Okay, so the convoluted means coiled. They are highly coiled tubes. That's why they are called as convoluted tubules. They are proximal to the glomerulus. That's why they are called as proximal convoluted tubules. Okay, then these proximal convoluted tubules are further extended to form the loop of Henle. So this yellow colored loop is the loop of Henle. and this part is called as descending limb of loop of Henle, and the opposite is the ascending limb of the loop of Henle, because the filtrate will flow from the descending limb to the ascending limb. And this loop is the loop of Henle. Okay. Then the ascending limb is further extended to the distal convoluted tubule. As it is away from the glomerulus, it is called as the distal convoluted tubules, and then distal convoluted tubule is connected to the collecting duct. Now there is the blood supply to this nephron, and the blood supply is explained in this way, that the afferent arteriole takes up blood to the.
थोड़ा loudly बोलो.
Okay, sir. Towards the glomerulus, and efferent arteriole takes the blood away from the glomerulus. Then these efferent arterioles are further divided into capillaries that surround the whole nephron, and these capillaries are called as peritubular capillaries. And the part of the capillaries that provide the blood supply to the loop of Henle is called as the vasa recta. On the basis of the length of the loop of Henle, the nephron is divided into two parts. One is the corticular nephron and the other is the juxtamedullary nephrons. So the corticular nephrons have the vasa recta, which is absent or highly reduced. So the juxtamedullary nephrons are the nephrons in which the loop of Henle resides deep into the medullary region. Okay, so this above region is the corticular region, and the below region is called as the medullary region. Okay, so the loop of Henle is deep inside the medulla, but the cortical nephrons are present in the cortical region of the kidney. And the major role of the juxtamedullary nephrons is to produce the concentrated urine. Now, urine formation. So how basically the urine formation takes place? There are three main processes: glomerular filtration, reabsorption, and the secretion. So the first is when the blood comes from the afferent arteriole, it is divided into the capillaries, and then the waste product is filtered from the capillaries to the Bowman's capsule. This is called as the glomerular filtration. The second is the reabsorption. So the first absorption takes place during the digestion. So this is called as the reabsorption. So the constituents like glucose and amino acids they are very important. That's why we cannot afford losing them. So it is very necessary to reabsorb them. So it is the tubular reabsorption takes place by the peritubular capillaries. The third is the secretion. So when some solutes are reabsorbed, it is also necessary to maintain the osmolarity. That's why some solutes are again secreted into the tubes, and it is the tubular secretion. Okay, so the urine formation can be explained with this equation, that first the glomerular filtrate is formed with the help of the filtration, then some amount is removed because it is reabsorbed, that way it is negative, and some of the solutes are added with the help of the secretion. That's how urine is formed. Now the first is the glomerular filtration. So when the heart pumps, in per minute each ventricle will pump 500 ml of blood, and kidney reaches 1/5 of it, that is approximately 1100 to 1200 ml of blood. Okay, so this much amount of blood is filtered by the kidney every minute. and this involved this is very important, has been can be done by the three layers. Okay, so as we have discussed the okay, so there is a okay, so there is a blood vessel lining of the blood vessel, then the lining of the Bowman's capsule, and the third layer which is present in between is called as the basement membrane. Okay, so the endothelial membrane, that is the membrane of the blood vessel, it prevents the filtration of the blood cells but allow all the components of the blood plasma that will flow through it, because there are the pores present in it. The second is the basal lamina, or we can say that basement membrane, through which only the large proteins can be prevented to filter out. The third is the slit membrane, which is formed by the pedicels of the podocytes. Podocytes are the cells that form the pedicels. These are the pedicels, and it form the filtration slit, and it is very thin slit, so that very small uh, substance cannot pass through it. That's why it is a very this type of filtration can be called as the ultrafiltration. A very high class filtration is here, and as we have seen that 1100 to 1200 ml of blood is filtered per minute, and out of which approximately 125 ml of filtrate is formed per minute, and this it is called as GFR. This is the amount of filtrate that is formed per minute by the kidney. That is the glomerular filtration rate. Next is that what is the pressure management in the Bowman's capsule. So the first of all, the glomerular blood hydrostatic pressure. So the cross section of the afferent arteriole is large, whereas the cross section of the efferent arteriole is slow. So it makes high pressure inside the capillaries, and it is around 55 mm of Hg. The next pressure is the capsule hydrostatic pressure. It is formed due to the capsular structure of the Bowman's capsule, and it is opposite to the glomerular blood hydrostatic pressure. Hydrostatic pressure, because this capsule formation, when this capsule is saturated with the filtrate, there is no space so that the filtrate can come out of it, and that's why it will move back into the blood vessels, and the pressure here is 15 mm of Hg. The third pressure is the blood colloid osmotic pressure, which is 30 mm of Hg, because of the colloid present in the blood that involves the blood cell, platelets, proteins, and etc. And here the blood colloids are very less. That's why the filtrate tends to move inside it, and this pressure is 30 mm of Hg. So the net filtration pressure should be uh, these both the first one is in the direction, so that the filtrate will move in the PCT, that is proximal convoluted tubule, whereas the second and third, they are the opposite pressures. That's why the net pressure is 10 mm of Hg, and then the filtrate will move towards the proximal convoluted tubule with the pressure of 10 mm of Hg. The next is the regulation of the glomerular filtration rate. Okay, so there is a process that is known as the autoregulation, that the nephron do not require any other organ to regulate the GFR, so it itself regulate it, because the as the GFR is increases, That means the water is increases, and also with water solute also increases. Okay, so there are two main cells that play a very important role in this regulation. The one is the macula densa cells and the other is the juxtaglomerular cells. So macula densa cells are present in the distal convoluted tubule. So after the uh, ascending loop of Henle, the distal convoluted tubules is present somewhere near this afferent arteriole, and they have the macula densa cells. These cells have the receptors and that sense the salt concentration. Okay, so when the salt concentration increases, it will give paracrine signal to the juxtaglomerular cell, and hence it regulates the filtration rate. Okay, so when there is any disruption in the homeostasis, and this causes the increase in the GFR, then macula densa sends the high amount of salt, and it input the via the paracrine signal to the juxtaglomerular apparatus, that and then the juxtaglomerular apparatus decrease the nitric oxide secretion that is responsible for the constriction, and hence it decrease the blood flow, and finally the GFR is decreased, and then the homeostasis is returned. And there are another type of cells which are present, which are known as the mesangial cells. The mesangial cells are the macrophage of the kidney. These cells are actually digest the protein accumulated in the pedicels, because some of the proteins are accumulated in the pores and they have to be removed uh, from this pores, so that the filtration will takes place. So mesangial cells comes out there and phagocyte the accumulated proteins. The next is the reabsorption. As we saw that the GFR is 180 liter per day, and urine release per day is only 1.5 liter. So why is this much difference is there? Because of the reabsorption. Because in the nephron, the 99% of the filtrate has to be reabsorbed in the renal tubules, and there are many active or passive absorption by which the tubular epithelial cells absorb these solutes. The active absorption is takes place of the substance like glucose, amino acids, and sodium ion, and the passive absorptions of water in the initial segment of the nephron, and also there is the reabsorption of the nitrogenous wastes takes place. To maintain this reabsorption, to maintain the ionic balance, secretion is also very important. So there is the secretion of hydrogen, potassium, and ammonia into the filtrate to maintain the ionic and acidic balance of the body fluids. Okay, so here as we saw that the absorption of the sodium, water, and other nutrients is very, and potassium are very important. And to maintain the ionic balance, there is the secretion of ammonia, uh, H plus ion takes place. And here we can see that the reabsorption of urea also takes place, and it is very important, which which we will discuss later. Now, this is the function of the tubules. Before study the function of the tubules, it is very necessary to understand the histological features of the renal tubule and collecting duct. So, what type of cells are present in different sections of the tubes? So, in proximal convoluted tube, simple cuboidal epithelial cells with prominent brush border of microvilli is present. This microvilli will help in the increase in the surface area for absorption and secretion. In loop of Henle, the descending and the ascending limb, simple squamous epithelial cells are present. These cells facilitate the diffusion of gases in small molecules like urea and water. In loop of Henle, thick ascending limb, simple cuboidal to low columnar epithelial cells are present that are helpful in the secretion, absorption, protection, and transportation of the molecules. In most of the convoluted tubules, simple cuboidal epithelial cells are present, which are again helpful in the absorption and secretion. The last part of distal convoluted tubules and all of the collecting tube have the simple cuboidal epithelium that is consisting of principal cells and intercalated cells. So there are these two type of cells play a very important role in formation in conservation of water and to maintain the homeostasis of the body. Okay, so now we will discuss the function of the proximal convoluted tubule. So as we discussed that the proximal convoluted tubule lined by simple cuboidal brush border epithelium, which increase the surface area for reabsorption. So in this tubule, the reabsorption takes place of 70 to 80% of the filtered water, sodium ion and potassium ion, 100% of most filtered organic solutes such as glucose and amino acids, 50% of the filtered chloride is reabsorbed, 80 to 90% of the filtered bicarbonate ion, 50% of the filtered urea, and variable amount of filtered calcium, magnesium, and phosphate ion takes place. And there is to maintain the ionic balance, the secretion of H plus ion, ammonium ion, and urea also takes place. So now we'll see how it is takes place. So first is the sodium and the glucose transporter or any other transporter symport. The two sodium ion are symported with the glucose. Okay, they entered into the proximal convoluted tubule cells. The glucose is entered in the peritubular capillary. So this is the tubule fluid in tubule lumen.
बहुत ही कम हो जाता है वॉइस, कभी कभी बहुत ही कम हो जाता है।
ओके सर, ध्यान
बढ़ाओ
रखिएगा।
बढ़ाओ, या वो लगाए हुए हो ना ईयरफोन?
यस सर।
हाँ, तो उसको सामने से अभी जिस साउंड आया वैसे करो, कभी कभी पूरा खो जाते हो जैसे।
ओके सर। So this is the fluid in the tubule lumen, and this is the peritubular capillary, and these are the cells of proximal convoluted tubule. So the sodium enters into the proximal convoluted tubule with the glucose. The glucose then enter into the interstitial fluid and then peritubular capillary with the help of the facilitated diffusion. That means there are some specific proteins are present for the transport of the glucose, and the sodium is then transported to the peritubular capillary by the sodium pump with the use of ATP. Okay, so this sodium and glucose is transported from the tubule lumen to the peritubular capillary. Next is the sodium reabsorption and the H plus ion secretion, how it is takes place. So it is dependent on the sodium hydrogen antipode. So when one sodium ion enters into the cell, one hydrogen ions move out of the cell. Okay, this sodium ion moves to the peritubular capillary with the help of the active transport with the help of ATP. As the concentration of CO2 is more in the tubule lumen and the peritubular capillary, it will diffuse inside the proximal convoluted tubule cells, and it is also be formed by the metabolic reactions metabolic reactions, and then the CO2 reacts with the water molecule. It will form carbonic acid in the presence of carbonic anhydrase. This carbonic acid will readily dissociate into H plus ion, which is again uh, can be antiported with the sodium ion, and the bicarbonate is then facilitated by the process of facilitated diffusion, is transported in the peritubular capillary. The next is the bicarbonate reabsorption. So the bicarbonate present in the tubule lumen, it reacts with the H plus ion, which are come out in the exchange of the sodium ion. It will form carbonic acid in presence of carbonic anhydrase and dissociate into H2O and CO2. Because of the high concentration, CO2 again enters into the cell, reacts with H2O, form carbonic acid, dissociate into H plus and bicarbonate ion. This bicarbonate then enter the peritubular capillary by the uh, facilitated diffusion, and the sodium ion will transported to the peritubular capillary by the active transport, and the other ions are simply diffused from the tubule lumen to the peritubular capillary, and water will transported with the help of the osmosis process. Now the function of the loop of Henle. So here the absorption is the minimum. There is 15% of the filtered water is reabsorbed, 20 to 30% of the filtered sodium ion and potassium ion is reabsorbed, reabsorption of 40% of the filtered chloride, and here the 10 to 20% of the filtered bicarbonate is reabsorbed, and the other ions are reabsorbed like calcium and magnesium in variable amount. So as we have seen earlier that 100% of the organic solutes are absorbed in the PCT, so they are not present now in the loop of Henle, and as the 80 to 90% of the bicarbonate are reabsorbed in the PCT, and that now in loop of Henle 10 to 20% of the bicarbonate is reabsorbed. So now in the loop of Henle all the bicarbonate is reabsorbed. So okay, so loop of Henle play a very important role in maintenance of the high osmolarity, as it has an descending limb and an ascending limb. So this is the descending limb, because the filtrate will move from upward to lower direction, and it is the ascending limb. So the descending limb is permeable to water as well as urea, and the ascending limb has two parts. It is the thin portion and it is the thick portion. The thin portion has the similar property like the descending limb, but this thick ascending limb is impermeable to water but permeable to electrolytes like sodium, potassium and chloride. Okay, so in this for the in the this sodium, potassium and chloride, okay, so how they are reabsorbed in the loop of Henle. So this is a very important symporters, sodium, potassium and two chloride symporters. So the sodium, chloride and potassium, they all of these three are imported together, as there are leaky channel are present for the potassium, so potassium move back into the tubule lumen. The sodium is absorbed in the vasa recta with the help of the potassium pump. The chloride are uh, mm, from the leaky channels, they are absorbed in the vasa recta, and the other cations like sodium, potassium, calcium and magnesium, they are simply transported with the help of the diffusion. Okay, so here we can see that two chlorides are enter here, one sodium enter here, but the one potassium is reversed back into the tubule lumen. That's why the interstitial fluid is more negative than fluid in the tubule lumen, and this negative charge will give the gradient formation for the formation of the concentrated urine. Now the early distal convoluted tubule. So the early DCT reabsorb the rest of the 10 to 15% of the filtered water, the rest of the 5% of the filtered sodium ion, and 5 to 10% of the filtered chloride. So most of the ions, water, they are all reabsorbed up to the early distal convoluted tubule. And here the major role is of the sodium chloride symporters. So what happens here? So this is the tubule lumen, okay, and here is the peritubular capillary. So there are sodium chloride symporters. So with the sodium chloride also enter into the cell, okay, and here there are sodium potassium pumps are present. So sodium will pump sodium enter into the peritubular capillary, and potassium will move into the cell, and the chloride that is entered into the cell with the help of the leakage channel, it enters into the peritubular capillary. So here the reabsorption of sodium and chloride takes place. Now the late distal convoluted tubule and collecting duct. So as we have discussed that up to the early distal convoluted tubules, all the essential solutes and water reabsorption takes place. So now what will happen in the late distal convoluted tubule and collecting duct? So it is the amount of water and solute reabsorption and the amount of solute secretion, it is vary depending on the body's need. Okay, so it is totally dependent on the body's need. If the body requires water, then the concentrated urine will form. If there is plenty of water present inside the body, then diluted urine will form. Okay, so 90 to 95% of the filtered solutes and waters have returned to the bloodstream. So that's why as all the solutes are reabsorbed, there is a conditional reabsorption takes place of sodium ion and water. So not all the time it is occur, only when it is required. There is a reabsorption of bicarbonate and selective secretion. That means not all the ions are secreted here, only some selected ions are secreted here, like hydrogen ion, potassium ion, and ammonium whenever they are required. There are two types of cells as we have discussed earlier, the principal cells and the intercalated cells. The principal cells reabsorb sodium and secrete potassium. So there are leaky channels present. When sodium enters through the leaky channels with the help of the sodium potassium antipodes, this sodium potassium pump, the sodium enters in the peritubular capillary, and the potassium present in the interstitial fluid, it enters the cells. Some of the potassium again through the leaky channels come into the interstitial fluid, and some will come into the tubule lumen. The next is the intercalated cells. Intercalated cells reabsorb potassium with the help of the diffusion and bicarbonate and secrete H plus ion. The reabsorption of potassium and secretion of H plus ion takes place with the help of the hydrogen potassium ATPase.